So, hi, everyone. I'm Scott Podolsky. It's my pleasure to introduce my dear friend and colleague, Carol Newton. Can you all hear?
Yeah.
Oh, wow. All right, even louder. As I'm sure many of you know, Carol's long played a leading role in bringing concerns of social justice and health equity, along with sophisticated trial design and implementation to global health, especially to the treatment of drug-resistant tuberculosis. Carol received her BA in political science and French from the University of Rochester before receiving her master's.
Hey, great job. We're already talking here.
And by 2001, her Doctor of Science degree from the Harvard School of Public Health in International Health Epidemiology and Ecology. She began working with Partners in Health in 1996, which she continues through today, and joined our department in 2001, where she's now Professor of Global Health and Social Medicine. Carol's also an Associate Epidemiologist at the Division of Global Health Equity at Brigham Women's Hospital. Carol led the effort to bring treatments for TB to low and low-middle income countries, as exemplified by her key role alongside her departmental and KH colleagues in several iconic and paradigm-shifting trials in New England Journal of Medicine articles on treatment of drug-resistant, extensively drug-resistant TB. At the same time, she's led the effort more generally to apply clinical trials to the evaluation of TB interventions and to shape global TB policy in accordance with such findings. At the intersection of all this, of equity and epidemiology, Carol's the co-PI of the endTB and endTB-Q trials. There are two multi-country phase 3 RCTs of all oral, shortened, novel regimens for rifampin-resistant TB. So with that, we'll get to hear from Carol, and then we'll have time for Q&A. So, Carol, turn it over to you.
Thank you, Scott, for that. It's really a pleasure to be here today and to talk a little bit about how 3 decades of partnerships of different varieties and stability have contributed to making significant advances in the treatment of drug-resistant tuberculosis. And so this was really going to be a talk about the overall endTB project and its components, with a brief dive into the history, and with apologies to Paul and to the historians in the room and to the students in the master's program. I am going to skip the imperialism, the slavery, the neoliberalism that I acknowledge fully are the real determinants of the conditions found in Haiti when PIH and Paul first went there in the 1980s, and in Peru when Jim and the team first went in the 1990s. I'm gonna start just with this, these, I'm sorry, terrible photos that I took myself from this book. We're working with Scott in the Countway archives to have access to some of these gems from our friend and colleague and Howard Hyatt's trainee, Mark Rosenberg. And this tells the story of one of the patients who experienced drug-resistant TB in Peru in the early 2000s. Very similar stories we saw upon arrival in the mid-1990s. They were living in informal settlements in urban Lima. They were driven there to escape the extreme violence between the Shining Path revolutionaries and the Peruvian government. And the population of Lima ballooned in the 1980s and 1990s with housing, roads, infrastructure, really unable to support the new people who are arriving with a lot of trauma from their experiences in the rural areas in Peru. They were, when diagnosed with TB, they were sort of confronted with the extremely rigid DOTS program, which was the approach, the global approach to simplify TB control. It had, the motivations were good. The motivations were to try to bring care to as many people as possible, but it was a very rigid program that actually aligned well with the values of the leadership in Peru at the time, Alberto Fujimori. It was somewhat authoritarian, but the heartbreaking experience of people like Joel Panmes revealed what happened when that very rigid, very limited strategy didn't work. And there was no backup and no acknowledgement that it could possibly fail. So several of Joel's siblings died of tuberculosis before he was diagnosed. He thankfully was cured, but you can see in the lower right-hand corner one of his quotes about the experience of taking the treatment that would ultimately cure him. So PIH did begin working in these informal settlements in Peru to try to begin treating drug-resistant TB, also in Haiti, and realized that just working in those places wasn't gonna be enough, and that it was a partnership really among PIH, among HMS, among the Brigham, that realized that the resistance to doing something bigger came largely from the global north, from the World Health Organization, from non-governmental organizations that had really invested in DOTS as the only solution. And so anything other than that solution was really rejected. I'm gonna pause right here because I see 4 seats open toward the front if any of the people who are standing in the back wanna come up.
Why not just encourage people to come through that front door if that's okay, Carol?
Yeah, fine with me. Actually, I'm gonna take this opportunity because I've got a little video clip set up and I have no idea if it's gonna work. Perfect. No, already doesn't. Apologies.
No, why is she not looking? Look at that. They were so over there. Live TV, am I right?
Sorry, everyone. So through this collaboration that I mentioned and really Howard Hyatt's, you can't hear this.
You can hear.
I don't really think I'm sustainable. Here, talk. People have epidemic.
I just wanted to share that clip. So Howard Hyatt helped us to convene a meeting at the American Academy of Arts and Sciences in 1998 that brought together kind of the global bodies that were influencing TB practices. And these were the kinds of reactions that these leaders had to the work that PIH and colleagues were conducting in Peru, that it wasn't possible to do in a framework of cost effectiveness, of only doing the bare minimum for those who were most impoverished. But out of that, despite that imperfect reaction, out of that came a temporary solution, something that was known as the Green Light Committee, which opened a very narrow door to providing broader access to treatment for drug-resistant tuberculosis in impoverished and marginalized populations. And it involved a collaboration with WHO and then with other non-governmental organizations, Doctors Without Borders, and interactive research and development to really begin this approach. That then translated into another collaboration with the Open Society Institute, with whom we here at the medical school, Meche led this effort together with Paul to produce this global impact report, which was really highlighting that drug-resistant TB was not just a disease of other. It was not just a disease that could be ignored, or if it was going to be ignored, it was at our peril. And it really raised the stakes, I think, for many of the people in the power centers in Europe. And also, had moved PIH into starting to treat drug-resistant TB in Tomsk, Siberia, together with colleagues there in the Department of Justice to work in the prisons, but also in the with the Department of Health. And then ultimately, this larger effort led to what was then the largest grant from the Gates Foundation, and it came to the medical school to really expand treatment for drug-resistant TB. So this is all happening in the late 90s and early 2000s. So these partnerships have this long foundation. So fast forward to 2012, 2013, we're still limping along with the same MDR-TB treatment of the 1990s, which truthfully was TB treatment of the mid-20th century. It was sort of mid-century modern, if you fancy that thing. It was really long. It was complex. It was toxic. There was a high pill burden. You saw the quote from Joël about taking 16 pills a day, and that was for 18 to 24 months, plus an injection every day for somewhere between 6 and 9 months. It was expensive, and the evidence was not super solid for what to do if you had choices between drugs. Nobody really knew what to do other than what had worked kind of anecdotally. And the benefits of this were very modest. So of the 500,000 new cases of drug-resistant TB occurring each year, only about 20% were even detected, diagnosed, and started on treatment. Only about half of those were cured. So you end up with literally 10% of the overall burden actually being treated successfully in this time period. So pretty grim for all the work that had gone into this. There was hope on the horizon in the form of these 2 new anti-TB drugs approved in turn by the US FDA, conditionally approved in 2012, and by the EMA in 2013, 2 completely new classes of drugs with new mechanisms of action, both based on phase 2 trials, just showing that the time to culture conversion, the time to convert sputum from positive for Mycobacterium tuberculosis to negative for Mycobacterium tuberculosis could be shortened by adding one of these new drugs to one of those long regimens I was showing you. So those trials were done. The companies got their at least conditional approvals, and in the case of Johnson & Johnson, they also got a priority review voucher, which at the time for the FDA, which at the time was estimated to be worth about $300 million to J&J. But there was no knowledge about how to optimally use these drugs and what we could do with them. Could they result in effective shortened regimens? So we took our experience in all of these partnerships and brought it to the endTB project to try to enhance the ability to access innovation cautiously and carefully, but to access it for those most marginalized and impoverished. So the endTB project launched as a partnership of a familiar group of players you see on the bottom, with the addition of Unitaid as the primary funder and a few additional research organizations. And it was really in response to the failure depicted by this, that I can't show you, the failure depicted by this image where about 1 million people were estimated to actually need treatment with these new drugs each year, and only about 11,000 were actually getting access to those new drugs. So the project was intending to close that gap and to use shall I say, a progressive interpretation of WHO guidelines to incorporate these newer drugs into routine care in larger numbers of patients. So the endTB observational study did exactly that in 17 countries, found about just over 2,800 people actually with multidrug resistant tuberculosis, and provided care using regimens comprising these new drugs and also some repurposed drugs. And then the endTB trial and the endTB clinical trial took the lessons from that first part and actually examined in randomized controlled clinical trials shortened treatments for two key groups of people living with multi-drug resistant TB. The endTB trial was those who were sick with bacteria that were susceptible to kind of the main drug used in drug-resistant TB, fluoroquinolones, and then the group that had disease caused by bugs that were resistant to that main drug. They were broken up into two different trials. This was an enormous effort. Unitaid provided $81 million over 9 years for these three studies and all of the dissemination efforts around them, plus additional co-funding was needed from PIH and MSF and funders to those organizations. So very brief summary of the endTB observational study, and many people in this room were involved, as Scott mentioned earlier, in all three of these efforts, just notably Molly Frankie in the endTB observational study. So it remains the largest observational study of regimens containing these newer drugs. It was carried out in 17 countries where either we or our partners worked. And the main results were that you could get much, much better effectiveness even with longer regimens. So more on the order of 80% compared to the 50% that we had been seeing before that. There were all kinds of probably, this is a separate talk, but all kinds of cautions about introducing these new drugs more aggressively because of fear of developing resistance. And there was a lot of tension around this issue with, again, many of the conventional global health authorities saying, whoa, you gotta be slow, you gotta be careful, and not really thinking about the people in front of them today who had terrible, terrible options. And so one of the areas of care was not mixing these two new drugs. They both had the potential for some cardiotoxicity, and rather than have an intentional approach to studying it, the advice was just don't use them together. So we were able to demonstrate that they were safe to be used together. They were safe to be used for longer than the labeled indication. And the data from the endTB observational study has resulted in more than a dozen publications. There's the QR code takes you to the endTB website, and you can see all of the publications if you follow that, if you're interested. They are all open access, so there should be no trouble getting access to them. And then the data were shared with the WHO to update guidelines, which relaxed some of the restrictions on the use of these newer drugs. So then now moving on to the second and third studies, the endTB and the endTB-Q trials. So the endTB trial I mentioned was focused on people with fluoroquinolone susceptible but rifampin resistant TB. So they couldn't get the standard 6-month treatment for kind of run-of-the-mill TB, but they could have regimens that contained the fluoroquinolones. And this trial innovated in many, many ways. Notably, it used a Bayesian response adaptive randomization. So it used accumulating information from the trial to change the number of people who would get assigned to each regimen. So more people would get assigned to the regimens that were performing better. And we were also really trying to find as many regimens that were effective and safe as possible. We weren't looking for a single winner. It is just not feasible to believe that the 500,000 new people with MDR-TB every year and the 500,000 prevalent cases of MDR-TB can all be treated with one regimen. And so we were looking for options depending on patient profiles and availability of drugs. It's important to note that the control arm was, again, I'm using the term progressive interpretation of WHO recommendations. So WHO might make a cautious recommendation about the length of using one of these drugs or combinations, and we opted to kind of go full force. And then another innovation was that where possible, we co-enrolled these two trials, the endTB trial and the endTB-Q trial. So there was a period when enrollment was overlapping and a number of countries and sites where enrollment was overlapping. And the only difference in eligibility criteria could be, in theory, could be determined by a rapid molecular test for resistance to fluoroquinolones. The endTB-Q trial was looking at a strategy of either a 6-month regimen for people who had less severe disease or a 9-month regimen for people who had more severe disease compared to this same progressive WHO recommended regimen. The primary endpoint for both trials was at 73 weeks, or about just under a year and a half. The big concern with these shorter regimens is relapse. And so we wanted to make sure that we could follow people who had gotten the shorter regimens for long enough to have a sense of the risk of relapse. Bless you. So speaking specifically and briefly about the endTB trial, just again, a reminder of what the design is, and I'll go through the alphabet soup of the regimens just a little bit. It's not gonna be, I think, super important for most of you. So all of the regimens contained one of the new agents, meaning bedaquiline and or delaminid. All of the regimens contained this first-line drug, pyrazinamide, because it had been very important to shortening treatment for drug-susceptible TB. So that was the prime rationale for including pyrazinamide in these regimens. They all contained a fluoroquinolone, which at the time was kind of the foundation of treatment for drug-resistant TB. And then they used one or both of these new repurposed drugs that did not have an indication for TB, but have been used off-label for TB for quite some time, to come up with a regimen of either 4 or 5 drugs, balancing the ideal bactericidal and sterilizing properties, also with toxicity risks. People were followed very, very closely for their entire time in the study, based weekly for the first 3 months, about monthly for the remainder of the first year, and every 6 to 8 weeks during the second year. And treatment support was provided, social support was provided in the model of PIH and the other NGOs, the service organizations that were involved in implementing this trial. Ultimately, the trial was carried out in these 7 countries. There were 754 patients randomized as participants in the trial. There was quite good distribution across regions of the world, so about 20% from Southern Africa, about 20% from South Asia, another 25% from Eastern Europe and Central Asia, and about 40% from South America, all in Peru, where they enrolled, as you can see, the lion's share of participants. It was a long haul. We started, the first participant was enrolled in Georgia in February of 2017, and the last participant was randomized in 2021. COVID obviously was no small factor in this trial. It was incredible what the teams on the ground did to keep the study participants safe was the first objective. On TB treatment was the second objective. And then in the trial was the third objective. There were no deaths due to COVID among trial participants, which is particularly noteworthy given that Peru had the highest excess mortality from COVID in the world. Okay, just some very quickly baseline characteristics in the endTB trial. This is aggregate. It's really hard, it turns out, to show 6 columns on a slide. So I don't advise a 6-arm trial for that reason. But there was very little difference in the distribution of these characteristics among the arms. Young population, which is what you would expect for TB, disproportionately male, also what you would expect for MDR-TB. You can see that there were important comorbidities in the population, so 14% with HIV, under 4% with either Hep B or Hep C, about 15% with diabetes. And these characteristics just show you that these were not a cherry-picked population of people who didn't have severe TB disease. We really wanted a population that was very generalizable. This was a phase 3 trial. We wanted these results to be meaningful for TB patients and programs. So this is a very quick summary of super complicated results. I've dispensed with the forest plots for this, but basically what you see here, the black column is the control arm. So again, it performed much, much better than what we had been seeing in routine care. And that was a really important attribute of the trial, that having a well-performing control arm was going to give us much more confidence if any or all of the arms were considered non-inferior to that control. Our definition of non-inferiority for the trial required that it be established in both a modified intent to treat population, which is kind of a more representative population, and in a per protocol population, which is really it's a reduced population where you're trying to eliminate the noise of factors other than efficacy. So it's kind of considered a purer measure of efficacy, but it's problematic despite that. So you can see, in addition to the fact that the control arm performed well, that 3 of the regimens, so-called endTB-1, endTB-2, and endTB-3, were all at least non-inferior to the control arm, and endTB was even superior to the control arm. Results were a little bit ambiguous for endTB-5, and endTB-4 was not non-inferior in any analysis. So basically we had 3 bedaquiline containing arms that were all non-inferior, but the bedaquiline sparing arms were not non-inferior. Again, summarizing a lot of data here just in one image on the safety of these regimens. We saw that people were followed very, very closely, and just using this one indicator of how many people had to have their treatment stopped because of toxicity, you can see that it was up over 40% in the control arm, but still in the 20 to 25% range in the non-inferior experimental arm. So certainly suboptimal, if better than what people were dealing with before. So the conclusions of the endTB trial were that we had this kind of transformation with 3 9-month regimens that were non-inferior to a very well-performing standard of care. There was really low mortality. Usually you see upwards of 10% mortality in cohorts of people with MDR-TB, and there was 2% mortality in this cohort. And this was not a cherry-picked population. It was a population that had cavitation on X-ray. It was a population that had some HIV as well as diabetes. We were a little disappointed about the toxicity profile. Again, an improvement, but not good enough. The liver toxicity is really the biggest issue, and that is associated with a number of the drugs across all of the regimens, including the control arm. The control arm also contains these drugs. And a big breakthrough, which was that these all 3 regimens could be used really in any age group during pregnancy, because they all had been endorsed already for use. The drugs had been endorsed already for use in pregnancy. And so this is the first time that young children and pregnant people could have shorter regimens using these newer regimens. So the endTB-Q trial, again, it's just going to be a rapid review. Enrolled 324 participants. Again, just to repeat, the strategy was kind of a stratified medicine strategy where people who had less severe disease got bedaquiline, clofazimide, delamanid, and linezolid for 6 months, and people who had more severe disease got those same 4 drugs for 9 months. Everybody was followed for at least 73 weeks, some up to 104 weeks. And the monitoring schedule was really the same. This is the distribution of enrollment for the endTB-Q trial, and also just to note that of the 1,030 individuals screened for endTB-Q, while 324 were enrolled in that trial, another 138 had been enrolled in the endTB trial. So this dual screening really allowed us to be more efficient in the processes, and also give people who wanted to participate in a trial a better chance of being in a trial. The geographic distribution is a little bit different. You can see much more heavily oriented to South Asia, with almost 80% coming from Pakistan and India. This is largely a function of the epidemiology of quinolone-resistant TB, where there's a higher burden in Asia than in other parts of the world. Also a long haul, the first participant was randomized in March of 2020, in the middle of the pandemic, by our incredible colleagues at IRD in Pakistan. And then we finished up last year, and these results are brand new. We just locked the database in October of this year and presented at the Union last month. Quick overview of the population. This time it is disaggregated by arm. You can see there aren't major differences between the experimental and control groups. I'll just note that there was a slight difference in the gender distribution, with a majority male in the experimental population, a majority female in the control. You can see, again, thinking about the comorbidities, less HIV in large part because of the geographic distribution of the participants, but more diabetes than we saw in the endTB population. And again, a group with severe disease where more than almost 75% had positive smears and 2/3 had cavitation on X-ray. Just one additional word about the progressive control arm. It was basically the same at its core as the experimental arm. So it was the same bedaquiline, clofazimine, delamanid, and linezolid, plus 1 or 2 drugs in 90% of the control arm, and it was delivered for 18 months in the control arm. So just something to bear in mind when you see the results. So here I am going to torture you with the forest plots. So this but I'll walk you through them. This shows you the relative efficacy of the regimens in the 2 analysis populations. So the MITT population, which again is kind of the more generalizable population, and the per protocol is a little bit more selected. And so you can see that, again, the control arm way outperformed expectations. It actually performed better than the control arm had with the fluoroquinolone in it, which was a huge surprise to us. It also outperformed the experimental strategy in both populations using our, I do have an advance, so I can do it here, right? This is our margin of non-inferiority, and the way you assess non-inferiority is to look to see whether the lower bound of the confidence interval around the effect estimate crosses that margin. So you can see that in the MITT population, the lower bound does not cross this margin, and in the per protocol population it does. So officially, we cannot consider that this regimen is non-inferior to the control that was tested. And I think really importantly, there were 8 people who experienced a recurrence of TB during follow-up. 8 people in the experiment, all in the experimental strategy, who experienced a recurrence. And this is something, had we not had a control arm, we wouldn't see this difference. So then that made us wonder, was there something that affected people differently? And what we have seen increasingly from other trials and observational studies in tuberculosis, drug susceptible TB, as well as drug resistant TB, is that shortening may not be for everyone, and that it really does seem to depend on the severity of disease. It might also depend on comorbidities. There's some evidence that HIV may reduce the relative benefits of shortening treatment. And so sure enough, when we looked at the distribution of favorable outcomes, we could see that they were not as good among people who had extensive disease and got the experimental strategy. You can see that among the people with limited disease, this forest plot tells you that actually the experimental strategy by point estimate outperformed outperformed the control arm. The confidence interval does cross zero, so there's some uncertainty around that benefit. But it's really the experience of people with extensive disease that's driving the difference between the experimental and the control arms. And then again, just to highlight, when we looked at recurrence in this population, 7 of the recurrences, I have this backwards, I'm so sorry. 7 of the recurrences were in people with extensive disease, 1 of them in a person with limited disease. And that was despite the people with extensive disease actually getting a little bit longer treatment. This is again a summary of the safety. As with endTB, little bit better in the experimental than in the control, but still not what I would consider ideal. Having 14.5% of the population having a serious adverse event during treatment or in the 4 weeks after treatment. And then you can see that there was more death in the experimental arm than in the control arm as well. So our conclusions are a little more cautious for the endTB-Q trial than for endTB. It appears from what we can ascertain that it could be a very good strategy for people with limited disease, but in extensive disease, it may not be adequate to prevent recurrent disease or relapse. We see how important the fluoroquinolone is in the shortening of treatment, and there have been other studies of fluoroquinolone sparing regimens in the last few years, and that have resulted in kind of quick recommendations for these regimens because they're much shorter than the current standard of care, and people are very enthusiastic about the results of single arm studies, for example. And it made us go back and look at some of those, and seeing that the relapse rate is actually very similar to what we saw in endTB-Q, but it seems to somehow just not not entering into the consciousness of people making policy decisions right now. So we have some recommendations for future trials, including the importance of an internal control, and that we would have celebrated the 86% success in endTB-Q if we hadn't had a control arm that showed us that you could do as well or even better with the longer regimen. And so when I first saw these results and we were beginning to process them, I was reminded of a conversation that I had with Paul in talking to him about the whole endTB project and our attempts to shorten regimens. And this is what he said to me. He's like, the objective is not to shorten for the sake of shortening. And you have to remember that. You have to remember that the objective is to provide the best care possible. And if that best care happens to be a longer regimen, then we have to figure out how to support people through that and make sure that we're not engaging in a race to the bottom. But we are in a space right now in the global TB community where that is exactly what's happening. This is a campaign that has been started by a group that's started as a public-private partnership funded by the Gates Foundation, and now is more on its own. And they're running this 6 months max campaign because they have found regimens that perform well at 6 months, but they're essentially ignoring the very same relapse that made us have a more cautious view of the results.
Quick question, just tell us what happens with relapse. Do you treat again, or?
You have to treat again, and the really important question with relapse is how much new drug resistance has emerged. And we don't have complete results yet, but we're seeing quite a bit of new drug resistance emerging. So it's kind of it's the worst case scenario. You have people who already have what's considered pre-extensively drug resistant TB, and then they end up with extensively drug resistant TB. So this leads us to what else are we doing? So one of the areas where we've been working a lot over, really, again, in partnership over the last 20 or so years, is on the price of these newer drugs. So the top, the magenta, is the trajectory. Actually, I'm going to start with the bottom. Sorry, just kind of mustard is the trajectory of the price of bedaquiline. And you can see that it started at $900, and over the course of 10 years, it is now at $130 just as of last year for a course of treatment. Not at all coincidentally, the primary patent expired last year. The trajectory of delaminid has been much, much slower to come down. The primary patent just expired in September or August of this year. And there is now one generic manufacturer that has responded to a tender by the Global Drug Facility. And so it's gone from $1,200 to $800 still. The price is still way too high and still way over. Oh, I lost my target. Oh no, there's the target price. Sorry, the target generic price there is this red dashed line, and its calculations if manufacturing processes are optimized, what these drugs could cost. So you can see bedaquiline is almost there, and delaminid is nowhere near where it could be based on engineering experts smarter than I. What this means for the price of current regimens. So you can see that 2 of the endTB regimens are under what has been set as a target for programs of $500. The 6-month, another 6-month 4-drug regimen is also under that target, but basically anything that contains delaminid is still well above that target and way more than what they could be if conditions were optimized. Almost done. So some of the other things I just wanted to talk about that we've been really intentional about in this partnership is the process of dissemination. So this is just a picture from a face-to-face co-investigators meeting we had with people from all over the world last year, when we had first completed analysis of the endTB trial results, and we sat with communications people to talk about how do you communicate to lay audiences? How do you communicate to ministers of finance? How do you communicate to different stakeholders about these results? We then presented them at the big TB conference last year, and we pretty quickly got them into a preprint. And I am so glad that we did, because we got the preprint was, I think, January of this year. And as I mentioned earlier to some in the room, the publication is still not, it is currently in press, but not out. We also availed of the communications machinery of the members of our partnership to talk about these results. So through HMS, we went to WHO with the results. They convened a guidelines committee meeting in June and then released recommendations that included 3 of the endTB regimens in August. Again, more partnership communication, reaching also the PIH audience. And then there were a lot of dissemination activities in country. This is the principal investigator in Lesotho. There was this huge media interest in the results of the trial because TB is such a huge problem in Lesotho, and the article is in press. The endTB trial is in press finally at the New England Journal, but not out. Then we have also engaged with new partners. So there is this online community of young people who are big followers of the YouTuber and young adult author John Green, who have been galvanized around issues of equity and access in global health and have found ways that they can actually act. So people were acting to try to persuade Johnson & Johnson to pledge non-enforcement of their secondary patents. And I have to say, this is one of my favorite leads of a press release. Johnson & Johnson confirms intent not to enforce patents, secondary patents for bedaquiline. So they didn't want to make it seem that they had been pressured into this decision. They wanted to make it seem as if they had made up their minds before. There had been this massive online campaign and really culminating 10 years of advocacy. 10 years of advocacy. This is another issue that we've been working on a lot, is the cost of these rapid molecular diagnostics. Again, there are estimates that these tools should cost under $5 per cartridge. That allows the company that makes them to earn a reasonable profit over the cost. And they cost anywhere from $8 to $50. They have a whole suite of diagnostics, Cepheid and their parent company, Danaher. And so we have been trying to get the cost down. We had a modest success on that last year. The endTB Now Act is before Congress. It has actually passed the Senate unanimously this year. It is an act that doesn't come with any appropriation, so it makes it a little bit easier to get it through, but it basically encourages a set of standards for tuberculosis funding and creates a path for improved funding for TB. There is still a chance that it might make it through the House this year, but it's not looking super promising this year. But the people doing this advocacy work are less disheartened by the change of hands come January than I am, so I like to talk to them regularly. And it is again, like anything else, it is about building relationships, and that a lot of legislators, like there have been a lot of surprises, like people like Lindsey Graham, for example, who has been a very staunch supporter of TB funding and of PEPFAR funding. Yeah. Marco Rubio, who is expected to be the Secretary of State, is apparently another legislator who's been a very strong supporter of global health funding.
Well, they're quoted as saying because it dampens revolution in places.
Exactly,
Like,
exactly.
I think Paul heard
Well, thank you.
Lindsay say that at a Bush.
Yeah,
dinner,
yeah,
right?
no, the
Like,
motivations
We'll take
vary. Granted, and that is part, I mean, this is a great conversation to have, like, the uncomfortable relationships that we have to develop or that we choose to develop in these spaces in pursuit of equity. Another effort is to try to, the Global Fund to fight TB, AIDS, and malaria has a designated split among those three disease groups. And although TB is the largest infectious killer of people, it gets the smallest share of Global Fund funding. And so there is an effort to try to equalize that. I think it's called 33%. And these QR codes should work if anybody wants to sign petitions or learn more about these initiatives. And then again, the Green Brothers have, they have businesses that support efforts for global health equity. First, the Maternal Center of Excellence in Sierra Leone, and now they've rebranded and launched a tea company, all of the proceeds of which are being used to support TB search, treat, and prevent activities.
The good store.
The good store, yes. So thank you for that promo. And then obviously the research is going to continue, and it's going to continue in partnership. So WHO came out with their rapid communication. As I mentioned, there are 8 regimens or 8 options offered for the treatment of multidrug resistant tuberculosis, and very little information about how to choose among them, very little evidence on which to base that decision. So we submitted a proposal to the US NIH earlier this year to try to get funding to work together with trialists and other leaders of other observational studies to be able to apply some modern epidemiologic methods to get a better handle on how to make those choices. Patient preference and improved safety and tolerability obviously are huge issues. In TB, we do not ask patients what they want, what they would like, what is a priority to them, in the way that in some other fields, in some other disease areas, they have been asked. And so trying to think more about how to do that. Courtney and Molly have both explored some work in that space or in those spaces. And then, this question about whether, when and if and how shorter regimens are an option for people with extensive disease remains an open question. And then how we can do a better job of preventing acquisition of drug resistance. So there's the biological side of it, where mutants exist in a population, and if you select for those mutants with a treatment, you're going to get more resistance. But there are also structural parts of this where, again, Johnson & Johnson and Otsuka had no obligation to develop or to support others to develop drug susceptibility testing in parallel with the development of the drug. So these drugs became available, and the solution that many had was don't use them for fear of developing resistance, rather than we've got to be able to test for resistance everywhere these drugs are going to be used, and quickly and efficiently and reliably. Tons and tons of people to thank. This is not a comprehensive list, but as you can see, the NTB project was an enormous endeavor across all of the studies and all of the other access and dissemination activities. And here's just a photo of the endTB-Q project teams from around the world. So thank you. To answer questions or engage in some of the discussion. Yeah, go ahead, Eric.
Wow. Having been a member of this department for a long time, I have to say that the work first on treating people with HIV and then figuring out how to treat people with TB and then MDR-TB has inspired my career and continues to, and it's amazing. The question that, and so thank you.
Thank you. No, thank you. We're all in this together. This is.
So my question is related to extensive disease, but it's about extensive psychosocial problems. So I'm wondering what was the effect, or were there even exclusion criteria for people who are homeless, people who are living in extreme poverty, people with alcohol use disorder, people with other injection drug use?
Yeah.
From my experience with HIV, so many of the patients who had TB had one or more of those situations going on. So were there data on that? And if so, how did it affect?
It's a really good question. I didn't go into the details of inclusion exclusion criteria. So we did not have specific exclusion criteria for any of those that list of things you just mentioned, for homelessness, for HIV or even disease severity, for mental illness, for substance use disorders. We had to work really hard with investigators to not de facto use those as exclusion criteria. There was one like discretionary criterion that they could lean on if they wanted to. But we really did, like, as each site came online, we worked with investigators to make it clear that the trial doesn't really do anybody a lot of good if we only know how these regimens perform in people who have no other challenges in their lives. Right. So there was I don't have the numbers offhand on the number of people who had substance use disorders, who had been homeless, who had been incarcerated, but they were present in that population, and people who were actively drinking definitely during trial participation. We had a couple of people who were imprisoned during trial participation, I think 3, and in 2 of those cases, we were able to coordinate with prison staff to keep people on treatment and in trial. So I agree with you. It's a huge issue, and it's a question to always ask when you're seeing results.
You don't really show dropout rates. Were there dropouts?
I didn't show them in this case. There were very few in endTB-Q, more in endTB. I don't know if anybody else here remembers, if Jackie or Emily remembers offhand. I can't tell you offhand. It was a lot lower than in routine care, again, because of all the support that was provided.
And sorry, if I could just comment also, please. So the social support that I think we also wanted to make sure, like, if participants had adverse events, if they had a new cancer diagnosis or something like that, gratefully, like, our project was able to help compensate the participants, and also, you know, participants that either had mental health illness or substance use or things like that, we made sure that there at least were referrals in place to either, if, like, for example, our partners in health sites, many voices for mental health, or we saw from in NTB, if lessons learned there, like where we could kind of increase our impact or referral process. So for participants in NTB-Q, working in Kazakhstan, like connecting them with NGOs that would give food vouchers or kits, diapers, hygiene, like extra things just to help out not only the participants, but sometimes their families. That really helped. And again, also in Kazakhstan, they partnered with like a cultural center that gave tickets to the opera or things just to, make them feel, I guess, more seen and supported that way. So I would say the component of that, regardless if you're in the experimental arm or control arms, like everyone received the extra support. And I think that really helped.
I would include that in the publications. I think that's, I'm sure you do, yeah.
Social support is, I'm gonna go with student union and then you next.
So you mentioned that in the COVID time, there was no deaths. Was it like,
Due to COVID. Just wanna be clear.
was it like, given that TB patients has less immunity, was it because there was extra precautions taken for this trial included patients?
I think it was more, I don't think there was any like protective effect of having tuberculosis or receiving anti-TB treatment. I think it was the support and so for example, when people were coming for study visits, once the first study visits got transitioned to happening at home. So people didn't have to leave their homes and it was the study workers who were traveling. And then as the participants started coming back to health facilities, they weren't taking public transportation, they were getting private rides. I think the circumstances of their lives and the risk of exposure was really changed. They got more social support that allowed them maybe to stay home more than people otherwise would.
You also had a younger, right? Wasn't your average age
Yeah.
32? So
Yeah, but in Peru, that did not prevent you from dying. So go ahead, Jean, you had a question or?
Yeah, so I also wanted to echo like amazing work from you, Megan, Jade, Molly, all along the way, other people in the department. It really is awesome to see like practicing at the Brigham, treating MDR patients, your work come in that way too, right? It's not just for
That's cool.
global health, but I had a MDR CNS case
Oh my
that I
gosh.
presented on and pulling all of your work over time. It was fascinating to present. That's one of my first questions. You excluded CNS patients, CNS TB, right? Or did you not have enough?
We did not explicitly exclude them, but again, that was one of the discretionary things that could have been used. So there were some people with extrapulmonary manifestations,
Right.
but not really
Yeah, because
severe.
it gets tricky. I mean,
Yeah,
taking
like
a
not
CNS
CNS,
MDR
not
TB regimens are
biliary.
very,
Yeah,
very tricky. And so it brings up my couple questions that I said, I told the division I'd ask you once we got to something like this. One is Moxie versus Levaquin, right? So for me, I always choose Levaquin or levofloxacin because QTC prolongation is supposedly lower. Did you run into that with Moxie, that there's
yeah,
QT interval complications more so than Levaquin-based regimens? Or?
so this is getting really into the weeds, sorry. And I just for those who don't know, I always have to offer this, I'm not a clinician. So these are the two fluoroquinolones that were used in the regimens. And as I had mentioned, the fluoroquinolone was kind of the foundation of MDR treatment, and nobody was paying attention to QT prolongation before the new drugs came around, where Moxie is used in clinical trials as a positive control for QT prolongation. So what we saw, we did see some QT prolongation, but not nearly as what people feared ahead of time. I think 2% or under 2% had a grade 3 or higher QT prolongation. They were disproportionately in arms that got clofazimine and another QT prolonging drug. But I say disproportionately, but we're still only talking about like 6 cases. So it's a little bit hard to say.
And the other benefit too is that when you do drug levels for quinolones, levofloxacin is easier to go from 750 to 500 because those pills exist in Moxie.
Right,
You
it's
know,
just
like
a-
weird. So
right.
for a lot of reasons, I always thought, why don't we focus on the Levaquin? But I guess that's
The trialists who are doing work in first-line TB treatment are much bigger fans of Moxie because of its better sterilizing
Interesting.
activity.
Okay.
Yeah, and because it can overcome resistance. The therapeutic margin for levo does not seem to be very large.
okay. The other question is about diagnostics. So like when you're doing future research stuff, again, the MGB example, our budget at MGB is higher than the GDP of like 50 countries, right? And we still, so for cases of TB, we can't do gene expert on anything but sputa. They won't let us do it on any other sample. We don't have ultra. We have to send to the CDC in Atlanta for
Wow.
resistance once. I mean, it's really bad. And so I'm wondering, it looks like it's much better at your global health sites than it is at MSB. How can we get some of that
Yeah.
approach
I
here?
think, I think access to expert may well be better.
Yeah.
But it's still a huge issue because of the price of the cartridges. So there's one cartridge for this technology that Jean is talking about. You can get a result within a couple of hours from a sputum sample. It's a closed system. It looks like a Keurig, like the coffee maker there. It's relatively easy to use. You can get a result on whether or not there's the bacterium that causes TB present, and then whether it's resistant to rifampin with one test. That test currently costs $8 if you purchase it through the Global Drug Facility.
But not ultra. It's the normal. gene
No,
expert.
it's ultra,
Okay.
yeah.
Okay. Wow.
Yeah.
So did you use ultra towards the end of the?
We
trials?
ultra,
Oh, wow.
we were using ultra when
That's
it
awesome.
became available. And then there's a second test for the fluoroquinolone resistance, which is still costs $15 through that same mechanism. So there's a lot less of that test being used, and a lot more either treating without information about quinolone resistance, or waiting for phenotypic
Which is what we have to
PST.
do here. Yeah. It's amazing.
But also it's worth noting, like the pricing, it's not like there is a single price that everyone in the world gets equal access
Correct.
to either. It's like certain countries get this reduced pricing. Other countries, even though the reduced pricing exists, their own country's tariffs like mark it up.
Yeah.
And so like in Peru, it's like $30 a cartridge, or a lot of middle income countries.
That's an issue with the distributor. That's the other
Yeah.
thing. So this
I mean-
concessional pricing that you're talking about, that is in theory available in Peru, if they can't wait the 9 months that it takes to get it from the Global Drug Facility, and they try to buy it on the open market, the distributor there marks it up a lot more.
I don't know, can in the US, can we get stuff through?
The GDF now.
Yeah, we can't, right? Like,
No.
because this
You
happened
have
with like
to be like-
isoniazid, it's like.
You have to be like
yeah.
global fund eligible, I think is basic.
I was actually told to sneak a
Yeah.
sample in. To,
But-
at
yeah,
night.
but I think it's like this-
All old school Paul style.
But there's this like issue with like middle income countries in general, like as countries transition to upper middle income countries, you kind of like fall off this cliff of like from when you were eligible for subsidized diagnostics and drugs and stuff, and like all of a sudden your country is expected to pay 10 times as much.
Exactly.
for things.
Yeah, with this Global Drug Facility, I think it's either being global fund eligible or being considered a high burden country that gives you access, even if it is a middle income country. But your point is well taken.
I'd love to hear from Miriam about what this will mean.
Me
Like,
too. Okay, I was going to talk anyway.
Great.
Thank you very much for the presentation and for the trial. Actually, it's informed quite a lot of programs on TB. I'm talking specifically from my experience in Sierra Leone. When we started MDR was 2017 actually. So we have been seeing patients with giving injectables for over 6 months and keeping patients in hospital for 6 months. And some of these patients don't even have muscle mass for injectables. So giving them an injection for every day for 6 months was quite challenging. Sometimes you don't have anywhere to actually, yes, it was that bad. So, and obviously the side effects as well with the hearing loss with the injectables was also very drastic. I could remember one particular patient who was a teacher, a young man, a teacher who had sudden onset hearing loss, and you would imagine after treatment, what would they do with their lives again? So seeing TB treatments evolve has been quite beneficial for our settings. And even without the final results, we have been informed by the endTB trial through WHO, and also it has informed a lot of our regimen changes. We have started, we dropped off the injectables, went into the long regimens. Now we started with the shorter regimens, and now the BPAL-M is over 80% of our patients are on those regimens. My concern, I always try to think, I think I discussed this with you at some point, was what happens after these regimens fail,
Right.
which is inevitable.
It's inevitable.
Just today, I got a report about one case that had a relapse after BPAL. And then we don't have in-country facilities for second-line DST. So they had to be sent to Antwerp. And then the results came back and the patient was bedaquiline resistant. So now we have to start going back to the old regimens, the injectables and everything like that. So that's just my concern about the newer drugs and the fact that we need a lot more efforts to go into regimens for TB and to preempt some of these resistant patterns. So, but overall, this has been really, really helpful. And thank you very much for all your efforts, the entire team, for the endTB trial. Thank you.
Thank you, Miriam, for all your efforts in Sierra Leone. Yeah, Priyanka.
Thank you, Carol. I just have a question. So in your conclusions, you wrote that it's eligible for all of the adults, children, and also pregnant women. But I was actually reading the on the website, and there in the inclusion criteria, you have included, I mean, your team is included that the inclusion criteria is also the participants who are willing to use the contraceptions or are in the so they do not get pregnant during the trial.
Yeah, as you know, being willing to use contraception and not getting pregnant are two different things. So there were like 20 or 22 people who became pregnant during the endTB trial, and then 8 or 9 in the endTB-Q trial. In any case, the eligibility or the fact that the regimens are endorsed for pregnancy, it comes from the fact that WHO had already made recommendations that each of these drugs could safely or relatively safely be used during pregnancy. So it's not from the trial results itself.
Okay.
If that makes sense.
Yeah.
Thank you.
In addition to that, is there anything in place to follow up the babies born?
There is, yeah. So we're following babies for a year after birth, and it's a good question. We have to check back in with Natalie on more follow-up on endTB. Our amazing pharmacovigilance team in Geneva, and Edna worked very closely with the pharmacovigilance team on that. So yes, there was something in place. To be honest, it wasn't particularly well defined, like what the regulatory recommendations are for following, babies. Just sort of looking for anything unusual in development or growth, but there could be lots of other reasons aside from being exposed to TB treatment. Oh, yeah.
I was just curious if you have a sense of how different NTPs are thinking about incorporating the guidance related to these 9-month regimens and the choice between that and whether to treat patients with the 6-month BPAL-M regimen, and how they're recommending that physicians make that decision, and whether they're sort of to your point about patient choice. And I don't know if you have a sense of that.
Thanks, Molly. So I didn't, Mariama referenced this. I showed one slide with the price, but I didn't talk a lot about the other alternatives. So at the same time that the endTB trial was running, there were another couple of trials that looked at 6-month 3 and 4 drug regimens. I mentioned that one was an uncontrolled single-arm study, and that was a 3-drug regimen BPAL for people with quinolone resistant TB, and then another was a randomized controlled trial for people that had either susceptible or resistant TB. So the 6 months there's no doubt that it's generally speaking more appealing to programs and to patients than 6 months when that's the only thing that's presented to people. Would you like 6 months of, say, a 4-drug regimen, or would you like 9 months of a 4-drug regimen? The best information we have is from our colleagues in Peru, where their the guidance, the in-country guidance, which takes a very, very long time to develop and then get through all of the approvals in country, laid the foundation for the introduction of one of the endTB regimens basically immediately. Basically, as soon as we presented the results to them, the fact that 40% of the participants had been enrolled there, and the fact that their guidance essentially created a placeholder for that, made them move very quickly to start using that regimen. And they also didn't have access to the drugs in the 6-month regimen at that time. So they started with one of the 9-month regimens, and then when the 6-month regimen became available in Peru, they started using that kind of as their first line, but they've seen a couple of cases anecdotally where they feel like there isn't a good enough treatment response. And so they're starting to adapt to say, maybe we'll use the 9-month regimens for people with more severe disease and the 6-month regimens for people with less severe disease. I don't think we're there yet. Like, I don't think we have enough evidence to make that decision firmly, but it's like sort of a logical choice. I don't know if you have any thoughts on what might happen in Sierra Leone.
Yeah, so for going to the shorter regimens, one of the big concerns we had was the extent of lung disease, switching from long regimens to the shorter ones. Most of our patients have very bad lungs, bilateral cavitations and everything. So the initial WHO recommendation for shorter regimens was that all patients with extensive lung disease were excluded from the shorter regimen and put on the longer regimens. The rationale being drug penetration, and other things like that. So now, and the new recommendation saying a 6 or 9 month regimen was good enough for these patients as well, was not very well digested by us and the clinicians. We were not very comfortable in putting our patients with very bad lungs on these shorter regimens. So we had several engagements and saw results from other places, and we decided to do it cautiously. So on a case-by-case basis, we decided to enroll people with very bad lungs on the shorter regimen, but paid closer attention to them and see if we can switch them or we need to swap them early. Fortunately, we haven't had a lot of issues with these particular patients on the shorter regimen, but we're just starting now. I think a lot more studies would have to be done for people to actually be confident enough of getting, like I said, we just got one relapse case a few days ago, and that's very worrying in itself to see that if we might have maybe switched to the shorter ones too quickly. So yeah, there's a lot of information that's needed, mostly from experiences with in other countries as well.
Carol.
Hi, Salman. I don't see you, but I hear you.
Can you see me now?
Yes.
Okay, hey. So Carol, it's so interesting. As I'm listening to this whole discussion, I'm thinking back to, and you know this well, like the late 1990s, early 2000s, where our Russian colleagues argued that everyone can't get the same therapy because of lung damage. And they had a scale, an X-ray scale to measure levels of lung damage. And exactly as was just described, they would look at change in X-ray dynamics after 1 or 2 months. And then based on that, they would decide whether to keep people on the same regimen or change them. And that was completely poo-pooed by the international community. They were told that they were on the Madagascar of science and all this kinds of stuff. And in actual fact, it turns out that yet again, they're correct from just what we're having this discussion, because it really would take being able to scale, to look at X-rays, being able to rate their severity, and then being able to make a clinical decision based on that, and then following them to make sure that they actually have a positive change in X-ray dynamics.
Yeah, I think that's a really good point, Salman. And as you also know, there was a lot of resistance to using X-ray at all during the DOTS era. Part of the oversimplification was to say all of the diagnostic attention should be on smear microscopy. Anybody can do it. You can put it on a slide under a microscope. Never mind that it only finds about 50% of people who have TB. That seemed to be an irrelevant point. And the argument was that those were the most infectious people. That was the logic. And so chest X-ray really fell out of favor, if you will. And it was also, I think, a favorite thing to do was to criticize things that were done in the East. I think we're a long way from knowing whether the scale they were using is the right scale, and probably
Yeah.
need to do more work to validate scales, validate timing, discern whether that depends on what the composition of the regimen is, right? How good the penetration is into lesions, in order to do a better job. But I think you're right, it's absolutely an important part of care moving forward and deciding what the right composition and duration are.
Thanks for the great presentation.
Thanks, Juan.
So just because we're out of time, and I was so fascinating, Carol, we could keep talking about it, but thank you so much for, totally inspiring, totally fascinating. Thank you.